Professor, welcome to the show and thank you for coming on. Could you talk to us about inflammation and why do we need it?
Okay, so inflammation is a natural part of your response to infection. So, what happens is if you get a bug, a flu, or a virus or a bacteria, they come along and the body detects them and then initiates in a first part of the immune response, which is the way the body tries to get rid of an infection. And inflammation is the first part of that process. It's also triggered by an injury. So, imagine you cut yourself, you get swelling, redness, etc., etc. That's the whole part of the body's response to repair yourself. So inflammation is really important for removing infection and for initiating a repair process. There's a whole bunch of familiar signs like pain, redness, swelling, etc. All of these things, and heat, and all of these things are part of the remove, response, heal process that inflammation is important for. So it's a really important mechanism to protect yourself against infection and injury.
So, when does it go from being helpful to harmful for us?
Yeah, and so the actual details of how that happens are sort of the subject of many, many, many research programs at the moment. Because if we actually understood the answer to that question, we would be able to target it with drugs and so forth. So it's kind of like the holy grail is how can we stop that process? Occasions when it happens are multiple, unfortunately. So, for example, as we age, we tend to enter a state of what's called inflammaging. And so, if you looked at my inflammatory mediators compared to yours, mine would be higher than yours because I'm older than you. And that we don't really understand why that happens, but it's clear that it does. As you get older, you get into a state of inflammaging. And we think that underpins, or certainly is a contributing factor to many diseases like Alzheimer's, Parkinson's, so forth. Worse by various genetic or hereditary factors, and also by things like diet and so forth. So there are multiple factors that contribute to that. But what is clear is that this process, chronic inflammation, now underpins many of the diseases of old age and many other diseases that we recognise as being common problems, certainly in the Western world, and it does differ across the planet depending upon a multitude of factors.
So, before we get onto your beautiful study, which I very much enjoyed reading, your lab focuses on the NLRP3 inflammasome, if I'm correct. Could you just break that down for myself and for our listeners? Because it's quite a mouthful in itself.
Yeah, it is. So actually, my lab actually is focused on studying the inflammatory responses to infections, and a whole bunch of what we call misfolded proteins, which would be amyloid beta and so forth. And integrated to that is a protein complex in the body called the inflammasome. And so one of the receptors that drives that activation of that inflammasome complex, which is literally a sort of a wheel of proteins that drive two outcomes. They drive inflammation, predominantly through a protein called interleukin 1 beta, and secondary to interleukin 18. And they also drive cell death of that cell, so it destroys the cell and takes it out of circulation. NLRP3 is a really interesting, what's called receptor or sensor. And that protein seems to now be linked to a number of chronic inflammatory diseases. And we don't fully understand what activates it, but we know there are a number of factors that do activate it, ultimately then leading into triggering the inflammasome pathway, as it's called.
So, has there been studies connecting this inflammasome to Alzheimer's, obesity, autoimmune diseases? Are there increasing evidence around this?
Yeah. So certainly to Alzheimer's and Parkinson's, also to obesity, chronic heart disease, and then and also actually to things like atherosclerosis. So there are many, many diseases that currently seem to link to NLRP3, and we're just sort of exploring the biology within that. So it certainly seems to be part of the chronic inflammatory process and potentially part of chronic inflammaging as well.
Are there other inflammasomes which are famous, let's say?
Yeah. So much of the biology in this area has required the sort of early biology work in mice, and it now appears that what happens in humans and what happens in mice is slightly different. So I think the jury's a little bit out. So there's an inflammasome called NLRC4, and there's one driven by a protein called NLRP3. They're all from the same family of receptors, which are defined by the NLR bit. the nod-like receptor bit. But it's unclear exactly which receptor is important for inflammasome function in different cells and tissue of the human body. So the output is the same. So if we understand inflammasome biology per se, that certainly helps us manage and understand the output. The precise receptor that's involved is probably going to be very important in therapeutics in the future, but that in humans is only just emerging, because if you think about it, a lot of the biology is done by removing the receptor, for example, from a mouse. Well, we can't do that in people, so we have to look at genetic studies, and we can do it in cells. Actually, that's what's really groovy about CRISPR-Cas9, is we can chop different proteins out of human cells and understand the biology, and that's proving to be very, very rewarding. But it's obviously much more complicated when you move something from a mouse into humans.
So before we get into the nits and grits of your study, just to sort of reiterate, the inflammasome is almost like a fire alarm system for your body, and that could be activated by various stress signals or molecules that we're still trying to have a good understanding of.
Yeah, that's exactly right. And it's kind of particularly interesting because the body actually makes a lot of effort to keep inflammasomes switched off. And so what we think happens is that in chronic inflammation, once you switch the inflammasome on, it keeps on trundling along and keeps on causing more inflammation. So
this sort of positive feedback
it's a positive
loop.
feedback loop, precisely.
It's really good that you mentioned human and mouse, because I think one of the cool things about your study is that you did sort of human in vivo data, and then you looked at in vitro sort of work afterwards. And so tell us about your fasting study. I think you did a better job of explaining it rather than myself, because I thought it was really cool and really neat.
Yeah, so I've got colleagues both in the NIH who are featuring that paper and also in Switzerland, and they've both done short-term fasting studies. It was very interesting. And so what they did was they took before blood samples, starved people for a period of time, usually a short period of time, then they fed them, and then they measured the lipids before and after and looked at what happened during the starvation period itself. And so what they then looked at was, okay, so what happens when you fast? How does that affect your lipid profile? And then what happens when you eat and what lipids are regulated? And they found obviously they found a whole range of interesting lipid changes, but the one that really sort of shone out to me was arachidonic acid, which I hadn't expected at all, because arachidonic acid is supposed to be associated with inflammation.
Yep.
And, we found this kind of really curious switch that when inflammation was high, arachidonic acid was low. And that really was very intriguing in this study. And so hence the reason we then went on to do cellular studies to try and work out what the mechanism was, because the inflammatory mediator they were measuring was interleukin-1 beta. And so when arachidonic acid levels were switching, it was sort of inversely or the opposite to the interleukin-1 beta levels, and we're going, okay, so does that mean that arachidonic acid is somehow affecting inflammasome activity? And that's why we're seeing this switch between inflammasome activity and what's happening when you're starving and arachidonic acid. So it looked to be a really interesting but unexpected switch that was regulating inflammasome activity.
So if I got that correct, during fasting, there was an increase in arachidonic acid, and that led to suppression of IL-1 beta. Just to break it down for all of us, is arachidonic acid a lipid?
Yes.
And is it found in the plasma of...
So it's, clearly it is, which is also interesting because I didn't necessarily expect it to be there. Normally, but this will all be to do with cellular breakdown. So arachidonic acid is a lipid that's found in the cell. What happens is you have your cell membrane. There are various lipids that are present in the cell membrane. And there's an enzyme called phospholipase A2, which is part of the turnover of lipids in cell membranes, and that makes arachidonic acid. And so clearly lipids are soluble and able to go through cell membranes, because cell membranes are pretty much lipids and proteins. And so I sort of hadn't really thought about this hugely. But when they were lipid profiling what was present in plasma, they were finding things like arachidonic acid was present. And I hadn't really thought about that conceptually, but it certainly appears to be there. And this will be because they can flow in and out of cells. So they were indeed finding arachidonic acid in the plasma, which to my mind was kind of intriguing and unexpected, but probably not to somebody who understands lipids, because I'm not a lipid biologist.
I think traditionally, I'm just based on my very small amount of reading, is that arachidonic acid has been traditionally known as a pro-inflammatory cytokine. And so here, these results almost insinuate that arachidonic acid is almost a modulator of inflammation. Yeah, please go ahead.
Yeah, no, and I think that's what's intriguing, right? So the what happens is that arachidonic acid, when it's made, then goes through a number of different enzymatic pathways which change what it is. And so one of them is through a pathway through an enzyme called cyclooxygenase, and that produces pro-inflammatory lipids called prostaglandins. And so that has been known about for years and years, and we take a whole bunch of anti-inflammatory drugs, such as aspirin, which inhibits cyclooxygenase, aspirin and ibuprofen, for example. So common over-the-counter, what they're called non-steroidal anti-inflammatory drugs, are things like aspirin and ibuprofen. And they inhibit this metabolism of arachidonic acid to prostaglandins. And that's super important because prostaglandins are important in pain and peripheral inflammation and a whole bunch of functions. So what we kind of assumed is that, okay, if you block one enzyme in arachidonic acid's metabolism, then surely it will just get channeled through to other enzymes that also break it down. And there's a complicated metabolic pathway which processes arachidonic acid. So, but we knew that arachidonic acid did accumulate, and then it sort of got processed by other pathways. So what our work was suggesting was that actually this arachidonic acid, when it's not going down the prostaglandin pathway, seems to be doing something else and seems to be immunomodulatory, which is super interesting. And I was working on prostaglandins a long time ago when I did my first postdoc, so my first post-PhD research position. And we kind of at the time talked a lot about, well, do we think arachidonic actually does anything? And I was in a massive prostaglandin lab, so it was a super interesting question at the time. And I'm not sure we necessarily had the tools, the sophisticated tools, because my colleagues that did all the lipidomic analysis, I mean, they're phenomenal, and the kind of sophistication in the analysis techniques has changed so much from when I was starting out. And so I don't know whether we would have had the sophistication to be able to work out if arachidonic acid was there and having any effects in its own rights. But technology has moved on, and now we can actually see it. And it's kind of quite satisfying to think it has a modulatory role in its own right. And it's not just an inert substrate that goes down different pathways. But I have to say it has caused consternation, because I did have a number of emails from colleagues who'd worked in the field for a long time saying, well, this is very odd. And I'm going, yeah, it is very odd. But we did study this very hard because I thought it was odd too from my
So does arachidonic acid also inhibit the NLRP3 inflammasome?
Yeah, that was the direct effect we found, and that was what was most interesting. So I was working with a really talented PhD student called Milton Pereira at the time, and he and I thought, let's have a look, let's see. And we thought that perhaps prostaglandins might activate this pathway, because we thought that'd be kind of cool if that happens. But in fact, they didn't do that at all. But Milton was so surprised, because what he did was he looked at cells, he activated the inflammasome, he looked at what lipids were produced. And he said, you know what, Claire, it's really interesting. He said, arachidonic acid is there. And I said, well, that shouldn't be there. And he said, no, it's very strange. And so he took a whole bunch of lipids and put it on the inflammasome. And he says, very interesting, Claire, arachidonic acid is actually switching off inflammasome activity. I said, really? Are you sure? So I made him do the experiments a few times. And eventually we go, well, both of us were surprised, but it was very, very clear that this was exactly what was happening. And we had to do those experiments very carefully, because, arachidonic acid is not a very stable lipid, so you have to handle it in the right way. And we had great advice from our colleagues who worked on lipid analysis, and they said, you need to do this, you need to do this, you need to do this, to technically make sure that we got those experiments right. So we did end up going, okay, well, actually, this is really interesting, and how is this happening? And so, of course, the rest of the paper was to try and work out the mechanism by which it was having its effects. But the net answer to your question is yes, arachidonic acid was indeed switching off the NLRP3 inflammasome, and only the NLRP3 inflammasome. It wasn't affecting other inflammasomes, which was particularly interesting.
I guess you did this in human-derived monocyte cells as well. So there's no point replicating this in mice, is there, in vivo?
We did it in mouse cells and human cells at the same time, actually, because we knew that we would get asked that question by the reviewers. So we did the experiments both ways. And in fact, we started out, because chronologically this paper took a little time to produce, and we started out in mouse cells because at the time we weren't experienced in CRISPR-Cas9, but by the time we got towards the end of the paper with another great PhD student called Jonathan Ling, we were then able to use our CRISPR-Cas9 cell lines where we'd knocked out NLRP3 and repeat those experiments. So we could do both sets of experiments in parallel. And that was very helpful in building this body of evidence to show what was going on.
Your paper mentioned the use of NSAIDs or aspirin. Could you talk more about that? It was very interesting.
Yeah.
Just to put it bluntly.
Yeah, so for me, what was really interesting, so I'd worked on cyclooxygenase in John Vane's lab, who won the Nobel Prize for discovering prostaglandins many years ago. That was where my first postdoc was. And so what we thought was, so if we inhibit arachidonic acid metabolism, presumably there'll be more arachidonic acid around in the cells. And so we'll see what happens. And so we said, okay, this is really interesting. So if we're inhibiting cyclooxygenase with aspirin or ibuprofen, and we're getting accumulation of arachidonic acid, this is then genuinely switching off inflammation by switching off the NLRP3 inflammasome. So what we have then is that if you give a non-steroidal anti-inflammatory drug like aspirin or ibuprofen, you're switching off pain and prostaglandin-mediated inflammation, but you're having a secondary effect to also switch off interleukin-1 beta production from the inflammasome, and that is having another anti-inflammatory effect. So we're describing a second anti-inflammatory effect for things like aspirin and ibuprofen, which was particularly interesting, actually.
So we got to be very careful because I'm not a clinician. But could that potentially sort of insinuate any sort of therapeutic benefit in the future of using NSAIDs for, let's say, chronic inflammatory sort of diseases, aside from just pain?
Yeah, the thing, so I mean, one should never take these things without consulting one's doctor first, obviously. And I'm a veterinary surgeon, not a medic, so I can't comment on human patients. But the thing with NSAIDs is that they do have a raft of side effects. So actually, what I think is interesting here is it's giving us a pointer to saying, okay, so we're switching off a bunch of inflammatory pathways. That's a good start. But what we're now discovering in some of the really exciting work being done by different investigators and different pharmaceutical companies is we know there are a multitude of inflammatory pathways. So how are the different pathways involved? And the idea with that being, so for example, there are NLRP3 inhibitors in clinical trials. We could be starting to use those. And so if we've got a patient who's NSAID responsive, non-steroidal anti-inflammatory drug responsive, but is not responsive enough, does that mean we need to start looking at NLRP3 inhibition in chronic inflammation? And that will then get us out of having to keep these patients on NSAIDs. And then we don't have the side effects of gastric ulceration, which is a massive problem in patients that are on NSAIDs for a long period of time. But we're actually targeting an anti-inflammatory pathway, which NSAIDs also affect. So I think what it's doing is helping us understand how NSAIDs work.
Yeah.
And then move towards a better targeted therapy for chronic inflammation, which would be good because NSAIDs have been around a long time and we know they don't cure everything, but we do know that they have some beneficial effects.
Yeah, and I think the mechanism of action of NSAIDs is still not that sort of concrete, right? And so we can utilize...
we know about the prostaglandin arm, right? And that is totally concrete, but there are some other interesting effects coming out that we know NSAIDs are having some other non-prostaglandin directed effects. And I think that is very interesting. But to my mind, it's the move of, okay, these drugs are okay at doing an acute inflammatory response, but what do we need to do to develop better treatments for chronic inflammation to avoid the side effects of NSAIDs?
I'm very big on biomarkers. Do you think arachidonic acid could be utilized as a biomarker of chronic inflammation, or, potentially a clinical study, let's say, would be to look at levels of plasma arachidonic acid in sort of patients with neurodegenerative diseases or any sort of chronic inflammatory disease? Do you think potentially there could be some sort of promise there?
Well, I think the trouble with arachidonic acid is it's horribly unstable and it's quite difficult to measure. So I'm not convinced that that's the case. But interestingly, so work that we've been doing with my colleague David Klenneman in the Department of Chemistry is one of the things that happens when you activate the inflammasome, you activate NLRP3, it then forms this enormous blob in the cell from an adaptor protein called ASC. And you can actually measure blobs of the adaptor protein ASC in the plasma of patients with different diseases. So we can see this in patients with Parkinson's and Alzheimer's and various other things. So that's a direct measurement of inflammasome activity. And I think that that as a potential biomarker will be very useful. So that's the direction we're going because that's very stable and very easy to measure, whereas arachidonic acid requires quite a lot of specialist kit at the moment. There may come a day when people are routinely profiled for their lipids, in which case arachidonic acid may become useful. But I think in the meantime, the actual measurement of the inflammasome ASC spec itself is probably going to be a quicker solution to answering that question.
Could you test the ASC hypothesis in mice if you were to, say, replicate, let's say, a new neurodegenerative model in vivo and go from there? And
we could, but we don't really need to because we can already find it in patients with Parkinson's and Alzheimer's.
Yep.
So job done, basically.
Yeah, no, exactly. I mean, that's the beauty of big data and clinical sort of work, which is
and
very
collaborative
nice.
work as well,
Yeah.
because we work across. I really like collaborative work, and it's been so really fantastically beneficial to work with people across disciplines and between countries and so forth, that you can really start to pull together all these interesting strands that allow you to answer these kind of questions.
Yeah, definitely. I'm very interested in fasting and caloric restriction. Do you think potentially, because with your study, the volunteers had 500 calories of food, then fasted for 24 hours, and then had 500 calories refeed. There's different types of fasting protocols. There's like main three. Do you think there'd be differences potentially in these fasting protocols with regards to what's happening on a cellular level?
So that's a really good question. And do you know what? Every time I talk about this work to anybody, which fasting protocol do you recommend? And I go, oh, that's way above my pay grade, guys. So I genuinely don't know the answer to that question. I would expect fasting per se, because in two different studies with slightly different protocols, we saw the boost in arachidonic acid and then the drop in interleukin 1 beta and vice versa. So I think that is clear. Whether or not time differences in fasting protocols would make a difference to how long, because I don't, the increase in interleukin 1 beta seems to be fairly fast after the refeed, and I suspect actually it's probably not going to make that much of a difference. I think you'll probably see the same thing, that while you're starving, your interleukin 1 beta will be low and your arachidonic acid will be high, and you eat and then your interleukin 1 beta goes up because you've shut off your arachidonic acid production. Actually, that seems to be the sort of loop. And there's really beautiful work done by my Swiss colleagues actually looking at that, the neuro switch actually that leads to the rapid production of interleukin 1 beta. So I think we've got a mechanism by which feeding triggers inflammation rather than anything else. And I think that's really, really interesting. But this will be data that my clinician colleagues will be able to explore over time, because I don't know the answer to that question, but it's a good one, but I don't know the answer.
Yeah, I mean, just as a sort of, let's say, a thought experiment, is because, I've been doing a lot of reading, let's say, a literature review on intermittent fasting, and there's differences in protocols between, many different studies. The methodologies aren't as concrete as one should be. And most of them just look at it at, let's say, a physiological level and not at a biological or cellular level. And so one of the great things about your study was that you were able to sort of assess that across scales, let's say, from physiology all the way down to cellular sort of a level. Do you think your study will sort of catalyze better, well-designed intermittent fasting or caloric restriction studies to really, prosper, let's say, the clinical side of it as well?
I mean, I think certainly this is the kind of work that my colleagues in both the NIH in the States and in Switzerland are thinking about doing, or are doing actually. These kind of studies are ongoing, because it would be really nice to know. So the work that we presented was done in healthy people. So imagine that you've got a particular disease state. Is there an optimal time for caloric restriction? Is there optimal protocols? I think all of those questions and the concept of maintaining or changing your health by particularly defined caloric restriction protocols is a really interesting idea. And you can do this now by measuring some of the plasma markers that we've talked about. I think the challenge is you have to be really careful depending upon what your health status is. So, for example, it would be very dangerous to embark on caloric restriction if you're a diabetic, because you've got a whole bunch of other more complicated regulating factors. So I kind of feel it's watch this space and see what my clinician colleagues come out with, because I think it will all come out in the wash, what's good, what's bad, what's recommended and what's not, from the kind of really elegant plasma measurements of lipids and inflammatory markers and looking at those things in tandem now, because my colleagues were already doing it, but I now think it becomes a really more pressing need to understand this better, because you're right. People do different things, and who knows what's the best. And it's very difficult extracting the science from the social media and various other things to understand. The difference between possible truth and fiction, I think, is difficult to understand.
Yeah, I think I'm really hoping that someone can move into looking at intermittent fasting in chronic inflammatory diseases and neurodegeneration in a very sort of well-controlled, reproducible manner. I know it's not as therapeutically enticing as, let's say, NLRP3 inhibitors, but it's such an easy lifestyle choice to make that could have profound health benefits on one. Where do you think this whole field might be going?
But I mean, I think that's an obvious thing to do. I think it's easy in one way,
Uh-huh.
but I'm not sure everybody would agree with you that psychologically it's that easy. So I
Yes.
think that's an interesting challenge, right? So if you've got the willpower to do it, it's easy. And I think intrinsically that actually is part of the challenge that we deal with, right? Is the obvious what we should do, right? And how to get to where we need to be. And obesity is a huge problem like that. And there's how much of this is psychological, how much of it is biometabolic, because there are clearly people who have challenges in this area. What can we practically do? And are there some simple things? So imagine there were more simple ways of maybe spacing out your meals that would allow you to get the benefits. And I think that is definitely feasible. And I think bolting the science into this, and this is the kind of things that everybody's thinking about. Can we help people to get over stuff or to reduce the likelihood of having severe inflammation by dietary modulation that's not too psychologically demanding? Because actually 500 calories is not very much, and that I think is okay. Well, if you could space your meals out between this period and then have a longer overnight period of time when you can reduce your inflammatory markers, then you could imagine that you would then be on a downward inflammatory trajectory. And I think that potential is very exciting, but I would say it's not that trivial. I've got quite a lot of willpower, but I've yet to manage to achieve a fasting protocol much as I might like to.
Yeah, no, I tend to fast on my own accord, but still I think the science is very sort of minimal, let's say. So I do it out of habit. One sort of out of the box question, that experiment that your colleagues at the NIH did with regards to fasting, if they did it with, say, processed versus unprocessed food, no fasting, just processed and unprocessed food, and they looked at, again, arachidonic acid, and they looked at IL-1 beta, do you think that's a feasible experiment? And would it be helpful and insightful to really understand the impacts of processed foods? Because I think similar to intermittent fasting, there's a lot of work on processed foods, but not very good studies categorically giving us conclusive, meaningful data.
Yeah, no, I mean, I think that's a great question, actually. And certainly, a lot of the work that my colleagues are doing actually comes from thinking about dietary manipulations and how you can help people and what you eat and can you persuade people to eat differently. And yeah, it's. I mean, the short answer to your question is yes. I do think this would be potentially useful, and I'm sure it's the sort of thing that's ongoing at the moment. I think the challenge that we all face is how do we help people have a cheap, plentiful supply of food that's good for you and therefore is easy, particularly in times when we're all pretty short of cash at the moment. And I think that's the challenge really, is how do we manage to generate and help people achieve a really good quality diet in a cost effective way. And I think that is the challenge. Processed food is very cheap. That's a problem, right? I mean, processed food is a long time has too much sugar and too much salt. We already know that before anything I did, we already knew that was a problem, right? And now you think about the lipids and what's in the lipids, and is the calorific part driven by sugar, salt, fat? Or are there easy changes we can make to achieve that? And my kind of thing that my colleagues are doing are trying to understand exactly those things and then comparing it in normal healthy people to people that are not and that have various metabolic disorders or various problems. And I think it's a very complicated subject which is really only now being tackled in a rigorous scientific way. And that's probably facilitated by the tools and the technologies that we now have available to us to do it. But it is an area that's ripe for really accurate measurement to try and understand what's going on.
Yeah, because I guess what I'm trying to potentially get at is the fact that processed foods, they're very cheap. We may not be able to get away from them. It is what it is, sort of thing. But if one were to design an experiment where both groups would have highly processed foods, but one group was fasting and one group wasn't fasting, would fasting almost mitigate the effects of having a high processed diet? And so it's not then when it comes to, let's say, a human or person sort of perspective, is that yes, you can have processed foods because you may have to, because that's something you cannot control because of economics, but what you can control is your diet with regards to fasting protocols. And I think that could be potentially an interesting question, and it sort of is inspired from that, I'd say, simple experiment, but that sort of fasting human volunteers protocol that you put in your study.
Yeah, no, I agree. I mean, these are all great questions, and being able to scientifically assess exactly what's going on and the consequences of it, I think, are a key to really understanding what's possible and what's not, and the real implications of different food matters and the impact that they have upon the way in which your body responds.
Your clinician colleagues will probably have a better answer for this, but based on the surrounding literature now on fasting, what's your take on it? Do you think there's enough data for people to embed it within their diets and daily life now? Or do you think we need more clinical studies and more mechanistic studies to better have confidence in sort of implementing it into our daily lives?
I think that's a really interesting question. I mean, obviously, we've just come out of the Muslim fasting season that everybody does once a year, and I think there's a real place for timed bursts of fasting. And I think there's a whole lot of data we could have by looking at people that do that on a regular basis. Whether you should do it all the time, I think, is a moot point, because there is some data now coming out of studies suggesting that, for example, if you fast all the time, then you can, and I don't mean as in never eat, but I mean if you follow a fasting protocol all the time.
Yep.
There's some evidence to suggest that you can get a loss of muscle, for example. You can get a loss of cardiac muscle, and that would be could be a major problem, right? And so I think we don't understand fully exactly what's happening to the body if you put it in a prolonged fasting situation. Short-term fasts, particularly for losing weight, seem to be very successful. And I think there's a lot to be said for maybe saying, oh, I'm going to fast for a period of time every year, or I'm going to fast maybe, 1 week in 3 or something like that. I think these protocols are the things that will be really exciting to see how they come out after time. But, obviously there are a whole bunch of people who do this regularly once a year, and it's interesting to really think about that data as a whole and how that compares to populations that don't, for example.
Yeah, there was a recent study, I think, in Nature Medicine or one of the Nature journals, and they did time restricted eating different protocols in obese patients or patients with cardiometabolic sort of disorders. And They saw a reduction in weight, which was a very good sign, but they didn't see a reduction in visceral fat, which was quite sort of interesting. But then it's sort of bringing in the question, if you were to play devil's advocate, if you are having this sort of weight loss due to intermittent fasting, is it purely because of caloric restriction as a variable, or is it because intermittent fasting is causing, the formation of ketones, which then, has its own sort of mechanism? And I think that's where it gets sort of difficult in sort of deciphering the mechanism of action with intermittent fasting.
Yeah, I think it's just difficult to disentangle the two, right?
Yeah, exactly. And whoever can solve that with a very good experiment, they have my respect.
Absolutely.
With regards to longevity, because as you was mentioning at the beginning, inflammation or aging is an issue. What's your take on fasting and, let's say, longevity and aging in the future? Like, is there promise? Is it all hype? Like,
No, I think that's a good question. That is something, certainly something I've been thinking about a lot as I trundle towards old age, which I'm in denial about. Yeah, so I mean, we know that there's a whole bunch of factors that help with managing aging. So exercise, for example, and very, use it or lose it is the exercise mantra. But it's true for your brain as well. Use your brain or lose your capacity. And I suspect that fasting and dietary manipulation will come into this, because if you can decrease your background inflammation, which is what you get with inflammation, then presumably you will have a healthier and better quality of life as you age. And I think that's a totally reasonable premise. How we get there and whether or not fasting is going to form a part of that, I think will be an interesting point, because as your metabolism changes and as you age, there is a tendency also to lose muscle mass. So we've just got through a discussion saying, if you fast, you may potentially lose muscle. That
yeah.
is not going to be a benefit. So I think there's going to be really interesting, hopefully what will happen is that we'll have really interesting studies that will come out of this, which will be backed by hardcore science, so we can really understand where we are. Certainly reducing background inflammation in people as they age is, I think, definitely going to be considered a good thing. It's just how we actually get there is going to be a very interesting question.
Just a quick question before we round off. In your study, what was the age of the volunteers who fasted?
So now you're asking me and I can't remember. It's the honest answer. But I think they were kind of young to middle-aged. I don't think there was anybody very old. And I think that is what I mean about extending that study out, right? It's going to be important to understand the effects in different age groups, different health groups, who it benefits the most and who it's not beneficial in at all. If that's the case, I don't know. But different age groups is going to be a key one to study, because as you age, there's no doubt about it, your metabolism changes, and that has a big impact on your health.
Yeah, I think even just looking aside from age, if we're even sort of being very picky and pedantic, looking at it almost as a personalized approach or genomic approach would be sort of quite interesting if there's a subset of people who are hyper responsive to intermittent fasting. And because I think that's the same with diet. I think everyone thinks there's a one size fits all with diet, but I don't think so. I think, everyone has their strengths based on their genomic background. But I think trying to understand what patients or what populations would be hyper responders or hypo responders to intermittent fasting would be difficult, potentially, right?
Yeah.
But would be very sort of interesting line as a research program to go down to.
Yeah, I think so. And I think that, it's kind of, we all think a lot about personalized medicine. Because in the West we have the luxury of thinking about that. I don't always think about it, because I think I work a lot of infectious diseases, and that's a different kind of ball game, right?
Yeah.
But I think there's a lot to be said for us thinking about that on a personal level. And some people find it easier to lose weight, other people don't. Some people find it easier to lose weight when they're young, and when they're older, they're not able to do that. And as your hormones change, your metabolism changes and so forth. So I think there's a whole personalized, but also life course understanding around what happens to metabolism, how does it change, and how should one adapt one's lifestyle to fit that in order to have a good quality of life, but also an enjoyable quality of life. Because I think, it's important that we age well, but it's important that we age well and happily. And so all these balancing factors then need to be thought out.
Yeah, I guess if you're fasting 2 days a week to extend your lifespan by, let's say, 1 year, is that needed?
And is that fun?
Yeah, and I guess there's a myriad of variables at play, which what makes science, beautiful but also complex and annoying.
Yeah. But at least we have the techniques to think about this
Yes.
properly. And I think to me, to my mind, that's the take home for me from the study is like, okay, this is actually really interesting. And we now have a myriad of techniques which allows us to quantify what's going on and working out what the evidence is to support different processes and different adaptations in one's lifestyle. And if one can provide people with a good reason to do something, then I think that becomes really helpful. But you need the hardcore underpinning science coupled with the kind of technologies that allow us to measure those things fairly easily. And so that I think is a good rationale that I take home from my study anyway.
No, I think your study is very good, very enjoyable to read, and I think the take-home message is that fasting is promising potentially and is on the rise, and it's something we need to look out for deep mechanistic evidence, so we could be confident in not just implementing it in our lives, but also our loved ones' lives who are probably aging and or battling any sort of disease. So there is promise, which
Yeah.
is good, and
Yeah.
that's exciting. And I think your study is that sort of first study in my mind that has really gone down in that sort of amount of depth and sort of mechanism to hopefully catalyze a ton of other studies with good hardcore science backed behind it.
Yeah. Let's hope so.
Professor, thank you so much. This has been super fun. And thank you for coming on.
It's a pleasure.
Awesome.